cell number 3 was a secondary spermatocyte. And you are not going to be asked to differentiate primary from secondary spermatocyte on a microscope. I want you to know they exist. I want you to know that one of them is diploid and one of them is haploid, but you can differentiate them. By the end of today, you might be able to, but I'm not going to obsess about that differentiation under a microscope. So I won't ask you to do that. You can just say it's a spermatocyte. So it's either a spermatogonium, spermatocyte, or our 4th type of cell, but 3rd one that you'll have to identify under a microscope is a spermatid. It's like it's like a toddler sperm. It barely moves. It is really dependent upon the cells around it to stay alive, but it begins to look like sperm rather than these individual nice spherical cells. And I do want you to be able to point those out on a slide. They're pretty easy because the round nucleus that you see in all of these cells gets smushed down into this like bullet shape, like a really sharp bullet. And that's really what's called the head of the sperm that's being formed. And so you'll see that. You'll see this totally different shape. It's a really dense, dark little nucleus. And that's how you'll know it's a spermatid. And then the last one are spermatozoa. These are the ones that are actually released from the seminiferous tubule. But they're not worth identifying because they're pretty easy to identify, but they're also, the way our slides are cut, the center of the seminiferous tubule is a little bit drained, and so there's often nothing there anyway. So don't worry about identifying spermatozoa under a microscope, but those are basically, so if these were toddler sperm, these are like adolescent sperm. Yeah.
Might you ask us the difference between early and late spermatids?
No. I mean, no. One's more developed than the other, but nothing that you need to worry about. So those are your 3 basic cells in the seminiferous tubule that you need to identify under a microscope. Spermatogonia, just the spermatocytes, and then spermatids in general. And then the 4th one is the sustentacular cell, also known as Sertoli cell, also known as nurse cell. It's quite the notorious cell. It has a lot of names, but it's a really important one because what it is doing is maintaining the blood-testis barrier. So you keep your open immune system, your humoral immune system, away from these cells. And the cells that do that are these sustentacular cells. They have other roles that they're still discovering. They help maintain the pH. That's why they got the nurse cell name, is because they take care of the cells. But they do maintain this blood-testis barrier. And the cells are huge. Under a microscope, all you'll see is their nucleus. They're around the edge, and they have a little like pinpoint nucleus. Or nucleolus, actually, is what you're seeing. A little teeny black dot in the center. And then there'll be a bigger, much lighter dot that people think is the cell. They think what they're looking at is one cell with a little nucleus. But what they're actually looking at is a big nucleus with the nucleolus, and the cytoplasm is everything in green that you see here. So it's this monstrous cell. It's like the blob. It fills in all the cracks and spaces. It forms the structure and the barrier for your immune system to stay away from those secondary spermatocytes.
And it extends all the way to the lumen?
Yeah, it's the main structure as well that holds, because these are like, if you imagine like these are like marbles that are all stacked on each other, this is sort of the rubber cement that kind of holds them together. It allows you to slowly push them through, but keeps a nice structure within the seminiferous tubules.
We're only going to see the nuclei around the edge there.
You'll see the nucleus
near the
and the nucleoli, and they will be around the edge, yeah. And they're pretty easy to pick out because they look really different than everything else. They have just that little teeny nucleolus and a really light nucleus. So that's it for spermatogenesis, and that's really all you're going to be looking at under a microscope for this week's material in male reproductive. So let's take a look at a couple of these under the virtual slide box real quick. There are 2 different colored slides in your slide box. One of them looks like this. It's an H&E stain. But the other one is white colored. It's more like, it's not totally white, but it has very little color because it's actually a stain that's meant for picking out chromosomes. And so it's really cool to look at. Look at both of them. I actually like the white one better because it makes it easier to see the chromosomes. Come into better focus. Huh. This is not as high a resolution as I would have hoped. But this gives you an idea of the whole seminiferous tubule here. This one right there. And so the spermatogonia are smushed up against the side here and here. There are two right there. Probably another one right there. So they're very dark nuclei, there and there and there, all around the edge. Now, if you look at these ones, they're a little bit bigger. They've got this speckled, kind of irregular nucleus. Those are your primary spermatocytes, much larger. And then just below them, there's smaller ones. And these are probably all your secondary spermatocytes. And so again, all you really need to be able to do is pick out the spermatogonia on the edge. The next about 4 cell layers are going to be spermatocytes. The big ones are primary, the smaller ones are secondary. And then eventually you'll get in here and you'll start to see nuclei that are kind of smushed down there, there, maybe there. Doesn't look like there's any over there, maybe that one there. Much closer to the center, those are your spermatids.
So where's the Sertoli on that?
So Sertoli, let's see, they do hide. That could be one right up there. Right there is one. So that is a nucleus, not the whole cell. That's kind of the crazy part. There's a little dark spot in the middle, it's the nucleolus, and its cytoplasm is just surrounding all the other cells, so you can't really distinguish it. But that's one right there. You have to look kind of closely, not that they're small, but because they're kind of light. So that's pretty much what you're going to be doing. So get some practice with that with both types of slides, and then you should be just fine. It's not too bad. All right, let's talk about female reproduction now. One last thing, spermiogenesis is on your list. Do not confuse it with spermatogenesis. Spermatogenesis is what we were just talking about. That's this process right here. Going from spermatogonia to spermatids. Well, really to spermatozoa, this whole process. Spermiogenesis is simply going from spermatid to functional sperm. And functional sperm have the head, this mid piece, which is full of mitochondria, and this tail, which is how this thing moves around. And so spermiogenesis has nothing to do with meiosis. Meiosis is done here. So this is really just changing the morphology or the shape of this cell, maturing it into a sperm. Most fertility issues in men are issues with this process right here, not fully going through spermiogenesis. Like the tight underwear thing, you know, temperature sensitivity. The enzymes that catalyze this process do not function at 96 degrees or above. And so if your testicles are warm all the time and they never get a chance to cool off, you'll still go through spermatogenesis, but you won't complete spermiogenesis. So you'll produce, some good sperm, but some pretty crappy sperm over here that are basically useless. And so they won't show up on sperm counts. And so that's the thing you're trying to, that colder temperature of the testes, which is controlled by the cremaster and dartos muscle mostly, you're trying to preserve the optimal temperature for spermiogenesis, not spermatogenesis. All right, now oogenesis. So similar process here, but instead of spermato or spermio, you're going to see oo because we're making not sperm, but we're making oocytes. And oocyte is just the name for a human egg cell. So on the left-hand side, this is another just simple drawing of meiosis. And this one now, for the first time, you can see the polar bodies being formed. When you're going through spermatogenesis, you started with your original cell that went through mitosis, and then one of those cells goes into meiosis. But with males, you had 4 cells created. Here, these are not cells, these little red ones. These are polar bodies because these are basically robbed. So you steal all the cytoplasm and you put it into our chosen cell right over here. So that's kind of how it's different. But again, the reason that the genetics are all the same, the inheritance pattern is the same, is because which one of these is chosen is totally random. 1 in 4 chance. It's not always the one on the right. Who knows? It's all random. So some of the names are going to be similar. So just like there was spermatogonia, there are oogonia. And these are our stem cells. So you're really just changing the prefix in the word. The oogonia turn into primary oocytes, just like primary spermatocytes. The primary oocyte then grows and turns into a secondary oocyte, but it takes a while. It's all the way down in a later stage that I'll talk about in a second. That's one difference, is that the secondary oocyte, you form the primary way up here, very much near the beginning, and then secondary is actually near the end of this entire process. But similar terminology there. There's some argument over whether or not secondary spermatocytes may show up earlier on. So you may read different sources that say, oh no, secondary oocyte actually shows up earlier on. We're going to stick with your book and what your notes say. So it says, on your notes, it says secondary oocyte in a mature follicle. You guys find that place on the page? You should go to that area anyway, because that's what I'm about to talk about.
Mature follicle, secondary.
Yeah, so secondary follicle has a primary oocyte, and it's not until what's called a mature follicle that has the secondary oocyte. So you may, especially, I know you guys don't use your lecture textbook much, but there's some contention behind whether or not that's true. But let's go through the difference between, the oocytes are just like the males, right? The secondary one is haploid, the primary one is diploid, same idea, just switch out put in oo instead of spermato before the words. And that's describing the egg cell itself. Now, in females, there's also a follicle around the oocyte. And there's a whole different set of terminology to describe the different stages of follicular development. Because sure, the egg goes through meiosis, but also the follicle grows immensely at the very same time. So it's very important not to confuse the terminology that we use to describe the oocyte with the terminology we use to describe the follicles. So let's look at the follicles. They're written on the right-hand side of this image right here. So when you have a primary oocyte, for males that would have been the end of the story. We got a primary spermatocyte. Great. Females, there's a primary oocyte, but it's packaged within a primordial follicle. You can barely see it here. The primordial follicle is just like a bunch of squamous cells surrounding a really big, so here's the oocyte, big oocyte. Here's the nucleus of the oocyte, and I'm drawing the primordial follicle now. Just these little squamous cells surrounding the whole thing. So that's what it's probably kind of hard to see in this one, but that's what's surrounding the primary oocyte at the very beginning. So primordial follicles, that's really the beginning of this story of follicular development. Starts with primordial, a simple layer of essentially squamous cells. It's not actually epithelium, but it really looks that way. So that primordial follicle hangs around for a very, very long time. These are all formed before birth. You actually just pause there as a primordial follicle and wait for follicular stimulating hormone to stimulate this follicle to change from looking like this, a primordial follicle. Once FSH shows up, those little flat cells turn into, we draw just a bunch of cuboidal cells that surround this cell. And once these cells are cuboidal, we call it not a primordial follicle, but a primary follicle. So they look at least like this. Primary follicles are going to get bigger, but this is like the smallest primary follicle, one simple layer of cuboidal cells. And again, it was follicular stimulating hormone that caused that change. Then the follicle is going to continue to grow because you're going to continue, it's a huge dump of follicular stimulating hormone that's going to cause this follicle to continue to grow. And so it's going to go from having a simple layer of cuboidal cells into a stratified layer of cuboidal cells, like 3, 4, sometimes even 5 cell layers thick. But it's still a primary follicle. And it's still just an oocyte inside. The primary oocyte hasn't changed yet. It's actually arrested in prophase 1 of meiosis. It's paused there. Then, eventually, the follicle grows large enough that it forms a little teeny crack around the oocyte. That's actually a pretty big crack in this illustration. But once the little crack shows up around the oocyte, the minute that little teeny crack shows up, we call it a secondary follicle. We're going to look at an example of this because that's probably the hardest one. I'm going to pull up a picture in a moment where we pick that up. But the distinguishing feature of a secondary follicle is that, yeah, sure, you've got a stratified follicle that grows, but then you get these little tears in it. And what it's doing is it's beginning to open up and form what's called an antrum. Once the antrum is fully formed, kind of like you see in this picture, we call it a Graphian or mature or vesicular follicle. There's a lot of names for this last follicle. I like mature because it's the last follicle. It doesn't get any bigger than a mature follicle. And again, that space in the inside is called the antrum. And it's huge. It's also like 5 times bigger than any other one. So again, we'll look at those two. Secondary, we'll look at all of these in a moment. But the differentiation between secondary and mature is probably the hardest one. You have a question, Morgan?
Probably you're about to show us some histology.
Okay, yeah, we'll look at all this and practice picking these out because it's, it's a continuum here. It's a growing thing, and we're kind of isolating primary, secondary, and mature. And those are a little bit hard to pick out, but we'll practice a couple times, and then it's not so bad.
So which one's the secondary?
So the secondary is not really illustrated in this image here. It would be right in here. It's closest to looking like this.
And that's when the antrum's starting to form.
There's just the very beginning of the antrum. Yeah, you're starting to sort of construct it, but once it's fully formed, then we call it the mature follicle. But I'll show you what fully formed means and what partially formed means in a second. And then eventually you're going to, once you are a mature follicle, that's actually when meiosis starts again. It was paused here in prophase 1. And I'm not going to ask you when things were paused. But it's an important thing because there's a difference in males and females when it comes to chromosomal abnormalities and numbers. I'm sure many of you have heard, you end up having more likely to have the wrong number of chromosomes after fertilization in an embryo if you're over the age of like 35, 36, and there are other factors that play a role, but age is a big one. Age is a really big predictor of things like Down syndrome. If you're 45, you're like 100 times more likely to have a child with Down syndrome than someone who's 25. And the reason is because you form the primary follicles before birth, and you go through this development while meiosis is paused in prophase 1. And it just has to wait there for like 30 years. It just sits there. Depends on when you get pregnant, but as much as 30 years, it's just waiting there, waiting to resume. And I'm doing this with my arms because you remember those like centrioles that come out and actually grab the chromosomes. They're holding them right there in prophase 1. And if they lose a grip, that's all it takes. And now when the cells split, they bring 2 chromosomes over, two things happen. One cell gets too many chromosomes and the other one gets not enough. Either one of those is a chromosomal abnormality. And so that's the unique difference between males and females. Males are just pumping it out. A sperm cell is not very old, even when you're 30. If you're a 30-year-old, the egg that you ovulate is old. It's an old cell, actually. Not to say it's bad or anything like that, but it's been around for a very long time. So those are the unique challenges. That's why males and females are quite different. But it turns out males, when they're about 55, also run into some significant chromosomal issues. It just happens to happen earlier in females. All right, so let's look at this under a microscope real quick. Ovaries are really cool to look at because they're relatively large. So here's a cartoon of an ovary first, just to give you kind of a caricature of a map. Our primordials, follicles with primary oocytes, are always on the edge. They're all over the place. There's like hundreds and thousands of these things. Then once one of them is stimulated by follicular stimulating hormone, it'll turn into a primary follicle. Now, in our slides, they're usually rat ovaries. Rats have a few more babies per, try than we do. So they're gonna have way more primary follicles, way more mature follicles, because rats can pump out like 9 babies at once. And so they're gonna have 9 mature follicles. They're gonna have a lot more than we do. So just remember that when you're looking at these. They're not human ovaries. But you're gonna have follicular stimulating hormone. Here's our secondary follicle here. This is a good representation of one. Primary, here's the beginnings of this antrum. Just these little cracks begin to form. Now we know it's no longer primary, it's now a secondary. Then here's a big, beautiful mature follicle right there. The mature follicle is what's going to eventually ovulate the oocyte, as well as the cells that are still attached to it, which is called the corona radiata, the radiating crown. Those are still attached to the oocyte during ovulation. So they're right here. When it's a mature follicle, they're just surrounding the oocyte, but you actually ovulate both the oocyte and the corona radiata around it. And that plays a role in fertilization because the sperm actually have to penetrate that. And so there's probably a lot more known about it it's probably much more important than most textbooks will talk about because I don't think we understand everything, but it probably plays a very big role in whether or not a sperm can successfully fertilize it. And then the second little detailed structure is called the zona pellucida. This one's kind of cool. It's easier to pick out in reality when you're looking at a microscope, but it is actually, so if we go back to this one, let's imagine these cells are now the corona radiata in the center here. And we're just kind of zoomed in at the oocyte here. Well, there's actually a thick protein shell that goes around just the oocyte. And this, a lot of things are known about this. Because it protects the cell, number one, it gives it a structure. Because the very, very large oocyte, it's hard to be a big cell. It's hard to keep your shape ideal, and this helps with that. It is actually kind of this color when you look under a microscope. Some of them it's a little bit more blue, but for the most part it's this dark pink ring. And it's all protein, it's not a cell, so there's no nuclei for what I just drew in. This is just a thick protein capsule around an oocyte. What does that sound like? Anybody eat eggs this morning?
Of the yolk.
So an eggshell is a zona pellucida that's calcified. That's all it is. And so certain vertebrates evolved to have ovulation happen with a hardened shell and then hopefully have fertilization happen before that, and then they got it out of their body, so they don't have to carry it around for a long time. They have to sit on it for a long time. So there are advantages and disadvantages, but we actually sort of have an eggshell. It's just not calcified. Instead, it's just protein. So it forms a somewhat protective layer, and again, that's called the zona pellucida.
So there's primary and secondary follicles and primary and secondary
Yes.
oocytes?
Yes. And in the secondary follicle,